Welcome to G Fun Facts Online. In the relentless war against cancer, we're recruiting some of the most unlikely soldiers imaginable: bacteria and viruses. For most of human history, these microscopic agents have been our sworn enemies, the cause of countless plagues and infections. But in a stunning scientific plot twist, researchers are now reprogramming these ancient foes, turning them into a synergistic, precision-guided force to conquer cancer. This isn't science fiction. This is the burgeoning field of bacterio-viral therapy, a strategy that promises to overcome the limitations of conventional treatments and write a new chapter in the story of oncology. The idea of using microbes to fight cancer isn't entirely new. It has its roots in curious observations made over a century ago. Let's travel back to the late 1800s and meet a New York bone surgeon named Dr. William B. Coley. Dr. Coley was intrigued by a patient with an inoperable tumor in his neck, who made a miraculous recovery. The reason? The patient had suffered a severe skin infection caused by Streptococcus bacteria. Coley theorized that the infection had somehow triggered the body's defenses to attack and destroy the cancer. He spent the next several decades trying to replicate this effect, developing a formula of heat-killed bacteria known as Coley's toxins. He injected this mixture into hundreds of patients, with some remarkable successes. However, the results were inconsistent, and with the rise of newer, more predictable treatments like radiation and chemotherapy, Coley's toxins faded into relative obscurity. But the seed was planted. That seed sprouted into one of the most successful immunotherapies still in use today, the BCG vaccine for bladder cancer. BCG, which stands for Bacillus calmette-guerin, is a weakened strain of the bacteria that causes tuberculosis in cattle. When instilled directly into the bladder after surgery, it provokes a powerful, localized immune response that mops up any remaining cancer cells. It remains the standard of care. On a parallel track, scientists were also noticing the anti-cancer potential of viruses. In the early 20th century, there were scattered reports of patients with cancers like leukemia or Hodgkin's lymphoma going into remission after contracting viral illnesses like measles or influenza. These early experiments were risky and unpredictable, but they pointed towards a powerful possibility that some viruses naturally prefer to infect and kill cancer cells. This field, known as oncolytic virotherapy, truly took off with the advent of genetic engineering. Scientists could finally take a virus and modify it, sharpening its cancer-killing abilities while making it safe for healthy tissue. A huge milestone came with the FDA approval of TVEC, a modified herpes simplex virus for treating advanced melanoma. TVEC is engineered to replicate inside tumor cells, causing them to burst. Crucially, it also carries a gene for a human protein that stimulates the immune system, calling in reinforcements to attack the tumor. The successes of BCG and TVEC proved that microbial therapies weren't just a historical curiosity. They were a viable, powerful strategy. But now, scientists are taking the next logical step. What if, instead of using them separately, we combine them? To understand why this is so revolutionary, we first need to grasp how each of these agents fights cancer on its own. Let's start with the bacteria. Bacteria possess a natural, almost spooky ability to target solid tumors. This is because tumors create a unique and chaotic internal environment. Their blood vessels are often leaky and disorganized, allowing bacteria circulating in the blood to slip out and accumulate within the tumor tissue. Furthermore, the core of a tumor is often a wasteland, low in oxygen, a condition called hypoxia, and filled with dead necrotic tissue. This environment is hostile to our immune cells, but it's a five-star resort for certain types of bacteria, especially those that can thrive without oxygen. Once they've set up camp inside the tumor, they can wage war in several ways. First, some bacteria produce toxins that directly kill cancer cells. Others simply outcompete the cancer cells for vital nutrients, effectively starving them to death. But perhaps their most important weapon is their ability to sound the alarm. The immune system is programmed to recognize bacteria as foreign invaders. bacterial components act like red flags, triggering a massive inflammatory response. This recruits an army of immune cells to the tumor site, breaking through the tumor's defenses and exposing the cancer cells to an attack they were previously hiding from. Modern science has supercharged these natural abilities. Using genetic engineering, we can now create what you might call bacteriobots. We can arm bacteria like Salmonella or Clostridium with specific therapeutic payloads. Imagine a bacterium designed to produce and release potent chemotherapy drugs, but only once it's safely inside the tumor. This minimizes side effects throughout the body and concentrates the firepower exactly where it's needed. Now, let's look at the viral assault. Oncolytic viruses are nature's own nanomachines, evolved to infect and replicate. The key to their therapeutic use is selectivity. We can either find viruses that naturally prefer cancer cells, or, more commonly, engineer them to be highly specific. For example, many cancer cells have a faulty p53 gene, a crucial tumor suppressor. We can design a virus that can only replicate in cells with a broken p53 pathway. In a normal, healthy cell, the virus is a dud, but in a cancer cell, it roars to life. The primary mechanism of attack is a process called oncolysis. A virus infects a cancer cell, hijacks its internal machinery, and forces it to produce thousands of new viral copies. Eventually, the cell becomes so full of viruses that it bursts, or lyses. This releases a new wave of viral soldiers to infect and destroy neighboring cancer cells, creating a chain reaction of destruction that can ripple through the tumor. But this cellular explosion does more than just kill one cell. It's an incredibly messy and loud event from an immunological perspective. The bursting cell releases a flood of signals that alert the immune system. It spills tumor-associated antigens, the very proteins that distinguish a cancer cell from a healthy one. It also releases danger signals and the viral particles themselves, all of which scream danger to the immune system. This process can transform an immunologically cold tumor, one that the immune system was ignoring, into a hot tumor, swarming with cancer-fighting T cells. This makes the tumor vulnerable not just to the virus, but to the body's full immune arsenal. So we have bacteria that can infiltrate deep into tumors and act as an alarm bell, and we have viruses that can selectively blow up cancer cells and expose them to the immune system. Both are powerful. But when you combine them, you create a synergy that solves the weaknesses of each therapy. One of the biggest challenges for oncolytic viruses is that our bodies are very good at fighting them off. Many of us have pre-existing antibodies to common viruses. If you inject an oncolytic virus into the bloodstream, these antibodies can neutralize it long before it ever reaches its target. This is where bacteria can play the role of a Trojan horse. By loading an oncolytic virus, or just its genetic blueprint, into a tumor-targeting bacterium, you can effectively cloak it from the immune system. The bacterium acts as an armored transport vehicle, protecting its viral payload. It swims through the bloodstream, homes in on the tumor's unique environment, and slips inside. Once safely within the tumor, the bacterium can be programmed to release the virus, unleashing it directly behind enemy lines. A groundbreaking example of this is a platform developed at Columbia University called CAPS-ID. Here, scientists use an engineered strain of Salmonella to carry the genetic material of an oncolytic virus. The bacteria are designed to break open only when they are inside cancer cells, releasing the viral RNA, which then starts the infection and oncolysis process. This strategy works even in animals that have antibodies against the virus, proving the effectiveness of the Trojan horse approach. But the synergy goes deeper. Solid tumors are like dense, poorly mapped fortresses. Viruses delivered through the bloodstream might only attack the outer layers. Bacteria, on the other hand, are motile. They can actively swim and crawl, penetrating deep into the tumor's hypoxic core, reaching cancer cells that other therapies can't. By carrying viruses with them, they ensure that the entire tumor mass, from the outside in, is targeted for destruction. They also launch a complementary immune attack. Bacteria are fantastic at kicking off a broad, rapid inflammatory response from the innate immune system. Viruses are particularly good at triggering a specific, long-lasting adaptive immune response against the tumor antigens they expose. Combining the two is like hitting the tumor with a 1-2 punch. The bacteria's initial inflammatory attack breaks down the tumor's immunosuppressive shield, creating the perfect opening for the virus-induced, highly targeted T-cell attack to follow. This brings us to a critical point: safety. Unleashing live, replicating microbes in the body sounds risky, and it is if not controlled. This is where the elegance of modern synthetic biology shines. Scientists can engineer a system of interdependence between the bacteria and the virus. In the CAPP-SID system, for instance, the oncolytic virus is engineered to be defective. It can't fully assemble itself into a new, infectious particle without a specific enzyme. And that enzyme is only produced by the carrier bacteria. This creates a beautiful safety lock. The virus can only replicate where the bacteria are present. And since the bacteria are designed to only thrive inside tumors, the entire therapeutic cycle is tethered to the cancer site. If any virus were to escape the tumor, it would be an inert dud, unable to harm healthy tissue. While most of this combined therapy is still in preclinical stages, the results are incredibly promising and are driving a strong push toward clinical trials. Regulatory agencies like the FDA are actively developing frameworks for these living medicines, learning from the experiences of BCG and TVEC. The journey is long, but a path is being forged. And the potential doesn't stop here. Researchers are already looking at how to combine bacterial-viral therapy with other treatments. The inflammation they cause could make tumors more sensitive to chemotherapy or radiation. But the most exciting prospect is combining them with immune checkpoint inhibitors. These are drugs that release the brakes on the immune system, but they only work on hot tumors that the immune system can already see. Bacterio-viral therapy is a master at turning cold tumors hot, potentially making checkpoint inhibitors effective for a much larger group of patients. Of course, significant hurdles remain. We need to perfect the delivery and ensure the microbes don't cause unintended infections or an over-the-top immune response called a cytokine storm. Manufacturing these complex living therapies at scale is another major challenge. And finally, there's the perception. The idea of being treated with live germs can be unsettling. Education and clear communication will be key to gaining acceptance from both doctors and patients. The future of this field is bright. We are moving towards next-generation engineering, creating smart microbes that can sense their environment and respond with tailored actions. We envision a future of personalized microbial cocktails, where a specific combination of bacteria and viruses is chosen based on a patient's unique tumor biology. The alliance of bacteria and viruses against cancer represents a true paradigm shift. By taking nature's most ancient life forms and augmenting them with the power of modern science, we are creating a therapy that is targeted, multi-pronged, and self-amplifying. The journey from Coley's serendipitous discovery to today's precisely engineered microbial systems is a testament to scientific creativity. The era of bacterio-viral synergy has dawned, and with it, a powerful new hope in our fight against cancer.